In this video, I'm going to show you how massive filters are processed in OnStock's lab at Princeton University. First, we sterilize petri dishes and 15 ml Falcon tubes by putting them under UV light for 20 minutes. Then, take a slice of massive filter from -80 freezer. One slice is usually about 2 cm thick. Let it defrost in a sterilized petri dish, which is placed inside a hood. The defrost process takes about 20 minutes. While waiting, prepare the following things: a marker, the sterilized Falcon tubes, a Bunsen burner, 70% ethanol, tissue towels, a tweezer, razor blades or surgical blades. Wipe the bench surface with 70% ethanol. Put labels on Falcon tubes: the sample name, the type of filter, outer membrane, massive filter, or inner membrane, and also the quantity of filters being put inside each Falcon tube. If you are using a razor blade, hold it with a tweezer and then dip inside 70% ethanol, and then flame sterilize them. Put them on the side of a petri dish cover. The red box indicates the area where you should put your fingers on to hold the blade, while leaving the other side for cutting. After 20 minutes, the massive filter is basically thawed. As long as it is not as hard as a rock, you can start working on it. The arrows shown in this photo indicates the water flow direction. Water flew from the outside towards inside. You can easily take away the plastic skeleton using your fingers. Cut the slice into quarters. Transfer one of the quarters to the petri dish cover, and then start separating the layers. Separate the fibrous outer membrane and the inner membrane from the massive filter. Cut them into smaller pieces. We first begin with the massive filter. The filter is highly folded in order to increase the surface area for capturing microbial cells. Cut every 2 to 3 folds. Make sure the cells are facing inside. This is to minimize the exposure area that will be in contact with the cutting tools. Make 4 to 5 cuts on each piece. Then you will have these strips of about 3 to 5 mm wide. Put them all inside one single Falcon tube. Place the tube in a -20 freezer for temporary storage until you have finished cutting the whole slice. Repeat the process for the outer membrane. Cut every 2 to 3 folds, make 4 to 5 cuts, and then put them into a tube. Repeat the same process for the inner membrane. Cut every 2 to 3 folds, make 4 to 5 cuts, put them into a tube. Place them in a -20 freezer for temporary storage. Repeat the whole process for the remaining 3 quarters. Label a Ziploc bag with sample name. Put the Falcon tubes inside the bag. Keep them at -80 degrees Celsius until you do extraction. It takes about 1 hour and a half to 2 hours to finish cutting a single slice of massive filter. Wear gloves during the whole process. Every now and then, spray your hands with 70% ethanol. Rub your hands against each other. Make sure the ethanol goes to your fingertips as well. Change the blade when the edge becomes blunt. Good luck on your cutting process.